Okay, this is lecture 2 of Mendelian patterns of inheritance. Basically, what we are going to look at is some genetic disorders, look at what autosomal and X-linked traits are, a little bit beyond the classical Mendelian genetics, and this will get us, if not to the end of the chapter, closer to the end of this chapter. Um, so what we have previously learned in the last lecture were monohybrid and dihybrid crosses. We also looked at a test cross and revisited some of those genetic terminologies such as genotype, phenotype. When you're looking at the genetic makeup of the gene, the genotype, then you can look at the alleles, which are functional units of a gene. That are alternate forms. You could say you have a homozygous dominant, homozygous recessive, or a heterozygous genotype. And if the principle of dominance applies, then the dominant allele masks the trait of the recessive allele. We've learned about the principle of dominance. We also looked at the law of segregation and the law of independent assortment. And basically what we've also learned is that if the genes are close to one another, especially genes closest to the centromere of a chromosome, those genes tend to segregate together. Basically, the determining factor of whether a gene segregates together with other genes or not would depend upon their map distance, or which would be the distance of where they are located on that chromosome. So Mendelian patterns of inheritance related to human disease. We know that genetic disorders are medical conditions caused by alleles inherited from parents. It's in this area where we do see some moral and ethical and some a little bit of controversy when looking at genetics and applying some of the modern day applications, molecular and genetic wise, in medicine for human beings. So basically what we have is autosomal genetic disorders are any genetic disorder or trait that is expressed on any chromosome other than the sex chromosomes, which are X and Y. When we looked at that karyotype cross, we know that chromosome pairs 1 through 22 are considered the autosome chromosomes, and therefore they would have these, excuse me, they would have these genes and would be considered autosomal related traits. So genetic disorders can be caused by genes on autosomes called autosomal disorders. Some genetic disorders are autosomal dominant. An individual with AA, capital A, or heterozygous would be phenotypically express the disorder. A homozygous genotype would not have the disorder. On the other end, if the disorder is autosomal recessive, anything with dominant alleles, such as a homozygous dominant or heterozygous genotype, would not have the disorder, and that disorder would only be expressed with a homozygous recessive trait. So what we could do is we could trace the track a particular disorder through a family tree. In genetics, we call that a pedigree. What we see here is we see a female represented by circles. We see a male represented by squares. The bond between the two would be a marriage, and then coming down from that, we'd have the children. So this particular, the first generation had 2 daughters who each married their husbands and had kids. The first couple in the second generation had 1 daughter. In the second generation, basically what we see here are 2 sons and a daughter. So this particular pedigree illustrates an autosomal recessive pedigree, and you can see wherever there is a homozygous recessive genotype, the allele is basically going to be expressed. The disorder is going to be expressed. So therefore, you would shade that entire circle or square in to express that it is being expressed there. So most affected children have unaffected parents. Heterozygotes have an unaffected phenotype. Two affected parents will always have affected children because they're always passing on that recessive allele. Close relatives who reproduce are more likely to have affected children. Both males and females are affected with equal frequency. So what you could see here is a special case scenario where the daughter of the one couple had a relationship with the son of the other couple, and they happen to be relatives. And what you could see there is that case of if you are a close relative who will reproduce, you're going to increase the chance of having an affected child. You could also have the case of an autosomal dominant and an autosomal dominant disorder. Basically what we would see here is the trait being passed through. So affected children will usually have an affected parent. Heterozygotes are affected. Two affected parents can produce an unaffected child, especially if they're, in the case of them being both heterozygotes. Both males and females are affected at equal frequency. So it would not really matter here. And you could see how that was passed through the table there. The pedigree, sorry. Mendelian patterns of inheritance in human disease. So autosomal recessive patterns of inheritance and disorders. If both parents carry one copy of a recessive gene, they are unaffected, but are capable of having children or a child with two copies of the gene who is affected. So that would be autosomal recessive. We do see that in two cases here. We see it in methemoglobinemia. It is a relatively harmless disorder, and basically what happens is you get an accumulation of methemoglobin in the blood, which causes the skin to appear a bluish purple color, giving it like that cyanotic appearance with which it would lack oxygen. The other case would be cystic fibrosis, holy lord. Mucus in the bronchial tubes and pancreatic ducts is particularly thick and viscous. What you see there, here you could see the case of methemoglobinemia. Here you can see how the hands appear blue compared to the red coloration that you would have here. And here's the case of cystic fibrosis, where you have those chloride ions and water are going to be trapped inside the cell. And because you have that defective chloride ion channel that does not allow chlorides to pass through, you get the lumen of the respiratory tract that is going to fill up with a thick, sticky mucus. Mendelian patterns of inheritance in human diseases. Such examples that are autosomal dominant patterns of inheritance and disorders. Two parents with a dominantly inherited disorder will be affected by one copy of the gene. If it is possible for them to have, unaffected children, however, in the case of a homozygous recessive genotype for that child. Osteogenesis imperfecta is one type of autosomal dominant disorder characterized by weak and brittle bones. Most cases are caused by mutation in genes required for the synthesis of type 1 collagen. The other one would be heredity spherocytosis, and it is caused by a mutation in the ankyrin 1 gene. Basically what happens there is the red blood cells become spherical, are fragile, and burst easily. So now looking beyond Mendelian patterns of inheritance, you should have an idea of multiple alleles. Some traits are controlled by multiple alleles, basically meaning that they have multiple allelic traits or allele types that will express the phenotype. The gene exists in several allelic forms, but each individual only has two alleles. Great example of this, again, would be the blood typing, where you have blood types, the alleles being capital I, capital A, capital I, capital B, and lowercase I, where the A is a antigen on the red blood cells, anti-B antibody in the plasma, and IB is the B antigen in red blood cells with an anti-A antibody in the plasma. And then I is neither A nor B antigens on the red blood cells, so both anti-A and anti-B antibodies in the plasma. The ABO blood types is an example of codominance. Basically, what you see here is that you have more than one allele is fully expressed. So both AI and AB are expressed in the presence of the other. So here we have the phenotypes of blood. Here you see that case of codominance where both alleles are going to contribute to the allelic frequency, that phenotype. So here are the different genotypes that give you blood type A. Here are the different genotypes that would give you blood type B, the genotype that would give you blood types AB, and the genotype that give you blood type O. I would learn these alleles and the phenotype that would be expressed based on crossing various parents. Codominance is also, if you go back, because you're contributing both. We also looked at this sophomore year codominant trait when we looked at chicken feather coloration, where you had the erminate chicken feathers. If you mix a black chicken with a white chicken, or not mix, but breed a black chicken with a white chicken, you get black and white speckled feathers. They are not gray feathers. They would be speckled black and white. That would be the case of erminate. The other case would be cross a red cow with a white bull, or vice versa, and what you'd have is a codominant trait where you get a roan offspring, where they have red and white hair. Incomplete dominance is when the heterozygote has a phenotype that is intermediate between that of the either homozygote. If you look at impatiens or 4 o'clock flowers, homozygous red has a red phenotype, homozygous white has the white phenotype, but the heterozygote would have the intermediate, which would be pink. The phenotype reveals a genotype without a test cross. And here you can see that there. So we are looking at the colors there. So R1, R2, R1, R2. If you cross, you get R1, 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 R2, R1, R2, R2, R2. And that will tell you the genotype and the phenotype there. So we'll do some practice of these as well. Continuing with that, you have human examples of incomplete dominance. Human examples of incomplete dominance would be familial hypercholesterolemia, or FH. Basically what happens here is the homozygotes for the mutant allele develop fatty deposits in the skin and tendons, which can lead to heart attacks during childhood. Heterozygotes may suffer heart attacks during early adulthood, and homozygotes for the normal allele do not have the disorder. Human examples of incomplete dominance, incomplete penetrance. The dominant allele may not always lead to the dominant phenotype in a heterozygote. Many dominant alleles exhibit varying degrees of penetrance. An example here would be polydactyly. Basically, the dominant trait itself is to have extra digits. So there are extra digits on the hands and feet, or both. So when we talk about digits, we're looking at our fingers and toes. So most of us express the recessive trait of that. However, the dominant trait would be having 6 fingers or 6 toes instead of 5. So not all individuals who inherit the dominant polydactyly allele will exhibit the trait itself. Pleiotropy is something we talked about before. This is a little bit beyond the scope of what you would need to know for the AP exam, but a good refresher. Basically what pleiotropic inheritance is it occurs when you have a single mutant gene affecting 2 or more distinct and seemingly unrelated traits. so it's found somewhere else. Marfan syndrome has been linked to a mutant gene that has been termed FBN1 and found on chromosome 15, and it codes for a fibrillin protein. Marfan syndrome is pleiotropic and it results in the following phenotypes. So you get disproportionately long arms, legs and hands and feet. You also get a weakened aorta and you have poor eyesight. If you look at Marfan syndrome here, you get connective tissue defects. You can see the skeletal tissue. So if you go back to connective tissue, bones, you could see the examples of what would happen to the bones. Blood tissue is in there. Eyes, lungs, and skin. You can see what happens there as well. So you get collapsed lungs, lens dislocate, severe nearsightedness. Polygenic traits occurs when a trait is governed by two or more sets of alleles. Each dominant allele has a quantitative effect on the phenotype. These effects are additive. It results in a continuous variation of phenotypes within a population. We do see this with skin coloration, height, and eye color. So these are all polygenic traits. The traits may also be affected by the environment as well. So if you look here, here you can see the parental generation crossing over. So here you can see how it's an accumulation there. The F1 generation, and then you have the F2 generation coming through, and then the proportion of the population. So 20 out of 64, 15 out of 64, 6 out of 64, and 1 out of 64. Remember, proportions can be converted over into percentages. So the percent of the population or a proportion of the population with these allelic frequencies and what that skin coloration will look like. So here would be a lighter tone of the skin, and over here you get a darker coloration of the skin. Then lastly, you do have X-linked inheritance. In mammals, the X and Y chromosome determines gender, with females being XX and males being XY. X-linked inheritance, the term X-linked is used for genes that have nothing to do with gender. X-linked genes are carried on the X chromosome. The Y chromosome does not carry these genes. It was discovered in the early 1900s by a group at Columbia University headed by Thomas Hunt Morgan. Thomas Hunt Morgan is well known. Mendel did a lot of work with pea plants. Thomas Hunt Morgan did an extensive amount of work with one of the most studied organisms in genetics, the fruit fly, Drosophila melanogaster. The other organism that has had a lot of research done on genetically would be a roundworm called Ascaris. So Thomas Hunt Morgan used fruit flies and he performed a lot of experiments to study X-linked traits. So he extended the basic Mendelian genetics and said, hey, look, not all traits are passed on via autosomal. Some of them are related to X linkage. They can be easily and inexpensively raised in simple laboratory glassware. This is referring to fruit flies. Fruit flies do have the same sex chromosome patterns as humans, so we could relate a lot to what happens with fruit flies to human studies. Morgan's experiments with X-linked genes applied directly to us as human beings. So if you look here you can see X-linked for eye coloration. If you look here, a wild type fly is one with normal wings, normal coloration, red eyes. That would be a wild type female in this case, being crossed with a white-eyed male. Basically what we see, because the female is homozygous dominant and the male is expressively white eyes, he is recessive there for the gene. All offspring are now going to have red eyes. However, over here, the female is a carrier. The male is not, because he does not have that second X chromosome. So he just has red eyes. But the female here is a female with red eyes, but a carrier for white eyes. And then if you were to cross these ones, here you could see the outcome in the F2 generation. So we have a normal wild type female. We have a red-eyed female who is a carrier for white eyes because she has the recessive allele there. We have a normal wild type male with a white-eyed male being in this square here. So you would need to indicate both the phenotype, the genotype with the phenotype, and specify the gender as well. X-linked inheritance, you could see it again going through here, looking at the gametes. So several sex X-linked recessive disorders do occur in humans. Color blindness, the allele for the blue sensitive protein is autosomal. The alleles for the red and green sensitive pigments are all on the X chromosome. Minx disorder is caused by defective allele on the X chromosome. Basically what happens is it disrupts the movement of metal copper in and out of cells. Phenotypes include kinky hair, poor muscle tone, seizures, and low body temperature. Muscular dystrophy, you've all heard of that, causes wasting away of the muscle. It is caused by the absence of a muscle protein called dystrophin. You all have heard of adrenoleukodystrophy. It is an X-linked recessive disorder. It is the failure of a carrier protein to move either an enzyme or a very long chain of fatty acids into peroxisomes, where peroxisomes would then help break them down. Adrenoleukodystrophy, of course, if you don't recall, is the disorder that Lorenzo had, and we learned a lot about Lorenzo and Lorenzo's oil. Hemophilia is another X-linked disorder that is recessive. It is the absence or the minimal presence of the clotting factor known as 8 or clotting factor 9. An affected person's blood either does not clot or clots very slowly. We do see hemophilia is often used as an example within the royal families of Europe. Hemophilia is called the bleeder's disease because the affected person's blood does not clot correctly or doesn't clot at all. People with hemophilia do bleed internally and externally after injury. Blood transfusions or clotting factor injections do help with the disorder. But why do we call it the royal disease? This pedigree will show how in close relationships within the royal family, especially during the era of Queen Victoria, hemophilia was passed on. So Queen Victoria was the first of the royals to carry the gene, and eventually it was spread throughout the royal families of Europe through arranged marriages between English, Spanish, Prussia, and Russian royal families. So you could see that here. So here you see Victoria and Edward, who had a daughter, Queen Victoria, who married Albert. And basically you could see all carriers here, carriers being passed through. What you see is the hemophiliac males are all going through the pedigree here, but what you see are carrier females that are coming through. So you can look at their pedigree and study the passing of that trait as it went through the lineage of the royal families. X-linked recessive pedigrees. Here you can see it. Grandfather was a carrier. Here we're looking at color blindness. So more males than females are affected. An affected son can have parents who have the normal phenotype. For a female to have the characteristic, her father must also have it. Her mother must have it to be a carrier. The characteristic often skips a generation from grandfather to grandson. If a woman has the characteristic, all of her sons will have it. We did see this with Lorenzo. That's it for genetics. We will do a little bit of practice of this, but we're going to keep moving on. We did go through this quite quickly, and we'll get through the next chapter fairly quickly. And then from here on out, it should be relatively new stuff. Genetics and molecular biology of a gene are what we finished with in advanced biology. So we will be getting into the new material very soon. I thank you again for watching the lecture. I hope you enjoyed it, and we will continue to move with great speed as the date of the AP exam nears closer. Thank you and have a nice day.